start with our nucleotides. There's a bunch of extra terms that float out there when we're looking at our genetic information. Almost always you'll hear people refer to them as the nucleotide. Right? There is nucleosides and nucleobases. They're kind of all looking at the same thing, sort of. They're like different stages of building upwards. So we could look at, say, an amino acid moving up to a tripeptide or a dipeptide, and we go from a tripeptide to a polypeptide. So you're moving through slightly different phases or different amounts of things being added to each of these pieces. That's really what's happening with your genetic information. When we polymerize your nucleotides, that's when we get our DNA and RNA. We'll look at some DNA aspects of structure, and then we'll look at the RNA classes. It says structural considerations. We aren't really looking at any structural considerations when we look at the RNA, okay, at least in this case. So, your nucleobases, all we're referring to when we're referring to the nucleobases, okay, has to do with kind of the ring systems that you're seeing here. Each of those are your standard nucleobases. And you may recognize these as your cytosine, thiamine, uracil, which we then go through and abbreviate, mainly because we can't say those words, C, T, U, G, and A. Okay, so those are our standards for our nucleobases. They get separated into two categories, purines and pyrimidines. Oh no, that is labeled right at the top. Sorry, it just looked a little weird. However you want to remember that is fine. And I don't think I actually directly build any test questions on that. So they're referred to as bases because if you take a look at your nitrogen within these, or a lot of those nitrogens, all of those nitrogens carry some basic characteristics. Okay? So your nitrogen is too sterically hindered in those cases to act as a nucleophile, but it does, each of those still has a lone pair. That lone pair can still be shared out with a hydrogen atom, and thus they're acting as a base. Okay, so that's where we're referring to them as our bases. The nucleo is probably coming from this information being found in the nucleus, okay, for the most part. So that's probably the origin of looking at our nucleobases. We can then scale these up to nucleosides. Okay, so a nucleoside is now taking a nucleobase and bonding it to a ketopentose. The most common ketopentose used is a ribose. Okay, so if we think back to our carbohydrate chapter, it's a keto because we're looking at the ketone functionality, or pent for our 5, and the os was ultimately just saying it was our sugar, or our carbohydrate, so our ketone for our ketone functionality. So that if we pull these out, that's actually interesting. Neither of those are ketos. Must be a typo on that. Both of those are aldos or aldehydes. These got to be aldopentoses. Okay, so we're looking at an aldehyde functional group. Where are we seeing that? How do we know these are aldoses and not ketoses? We find the anomeric carbon. The anomeric carbon, okay, most commonly is the one shown on the left, right? That anomeric carbon is what we're trying to identify as being an aldehyde or a ketone. Okay, how would we do that? Well, we push those electrons out, push those electrons down in all of these cases. Okay? Once we've gone through and made the nucleoside, we've lost that oxygen because the nitrogen is replacing it, but ultimately come back down, kick out. In all of these cases, what functional group do we end up with at the anomeric carbon? An aldehyde, not a ketone. Okay? So each of these are your aldoses. The two common ones that you see are your ribose and deoxyribose. Okay, what is the difference between ribose and deoxyribose? Deoxy meaning no oxygen. Okay? We could push that a step further, and if you look at the name on that, it says 2-deoxy, meaning at position 2, we've removed the oxygen. Very rarely do you actually see the full name drawn out. If you think to DNA, your DNA is deoxyribonucleic acid. What you're referring to is this overall process. Okay? That sugar has lost or has been exchanged a hydrogen or an OH for a hydrogen. Excuse me. When we're going through and doing that connection, you're linking your anomeric carbon to your nucleobase. Right? So that's that new bond forming there. We're losing that oxygen and we're replacing it with the nitrogen. That works fairly well overall because our nitrogen and our oxygen similar chemistries. We might predict that that's going to be something that's difficult to go through and exchange because is our nitrogen going to be particularly nucleophilic? Sterically hindered, that's going to be a little bit of a tedious process. So there's going to be an enzyme to help facilitate that linkage, make that a little bit cleaner and easier to work with. You can get into the real specifics on how they're connecting up to your pyrimidines and your purines. Okay. Did I label those right? I did label those right. You can say it's the first nitrogen or the ninth atom in that ring. Not that big of a deal. What we're really concerned about is looking at the overall structures. This then pushes us up to our nucleotides. All that's happening now is we've now added a phosphate. That's it. So our nucleotide is composed of a phosphate, your sugar, and the nucleobase. All three things go into that to end up with your nucleotide. Okay? They are named for the nucleoside plus the location of your phosphate group. Okay, so where is the phosphate group in each of these cases? So if we look at our sugar molecule, our anomeric carbon is number 1, 2, 3, 4, 5. So these are all 5' monophosphates because we've put the phosphate at that 5 position. Why do they call it 5' and not just 5? But we could just as easily have attached the phosphate, or not just as easily, it's a little more sterically hindered. We could have put the phosphate out here. Then that would have been a 3'. So we still use the prime, and that's still part of our ring. Why do we refer to those as primes and not just the number?
Direction of like transcription and translation.
So it gets mentioned in transcription and translation, okay? But that's still referencing those positions. Why are those positions called prime? Okay, how many rings are in the structure? There are 2 rings in each of those nucleotides. Which ring is the phosphate connected to? Which ring is the higher priority? So the higher priority ring goes to your nucleobase. Okay, so it gets numbered the normal numbers, 1 through however many are in it. Okay, so in our pyrimidines, was it 1 through 9? Yeah, 1 through 9, and in our purines, it's the other way. Your pyrimidines, 1 through 6, and your purines, 1 through 9. Okay, so we're numbering these guys. Okay. Our primary numbers, or numbering sequence, I don't even remember how they add up, go through up here. So those are 1, 2, 3, 4. Is it 5, 6? I don't remember how they number them. Okay. Which ultimately, again, isn't important for you guys. But those are numbered according to your standard numbers. We now have a second ring, so we can't then call that or continuing your number calculation off of that because those aren't part of the original ring. Okay. So to reference a second ring or even a third ring, we start adding primes. Okay. So if there was a third ring attached to this, how would that then get counted? 1 double prime, 2 double prime, 3 double prime. You're just referencing it being on a second ring. So that's where the prime is coming from. It's just a way to differentiate where your phosphate is located. It's not located on the primary ring structure. How do we know it's the primary ring structure? That's what we're starting with. Okay. Typically, we put the phosphate on the 5 prime, and then that phosphate can polymerize up to the 3 prime of another nucleotide. So if we look at our DNA primary structure, it's now just going to be the sequence of the nucleotides all the way through it. Okay? Each of those nucleotides will be connected through glycosidic bonds. Your glycosidic bond being out here. And then you've got, so glycosidic bonds, sorry, let's highlight that, to your nucleobases, and then you've got phosphodiester bonds going through the backbone. Okay, those are your phosphodiesters. And your glycosidics, not shown very well with their little arrow, is that red guy in there. Okay. If you were trying to go through and look at these, you may, as a differentiation between DNA and RNA, for instance, if we try to look at this sequence, we've got thymine, guanine, cytosine, adenine. That looks kind of sloppy, but it's close enough. Okay. If I want to emphasize which nucleotides are involved in this, I now need to add extra information. Because if we look at this, are those RNA or are those DNA? TGCA. I just look at that sequence. Ignore the T. GCA. Is GCA DNA or RNA? Could be either. Okay. So what we might add into that as extra information so that we know, hey, this strand that we're looking at is in particular DNA, not RNA. You may see a D included in front. Okay, lowercase d, standing for deoxy. Okay, that's it. We're just trying to specify that extra sugar. Yeah, not particularly anything else interesting on that. After that, we can move up to our secondary structure, and it's how these individual strands or polymer strands then coordinate to other strands. Okay, this was our famous experiment with Watson and Crick, and Rosalind Franklin actually, and yet another name that, anybody heard of Chablis?
Is it Watson and Crick the ones who stole it?
So there's a big debate over that. Rosalind Franklin was the one that did the X-ray crystallography. Watson and Crick allegedly were the ones that interpreted her work to come up with the double helix. Depending on whose story you buy, you're going to run into different arguments on that. This was happening, what are we looking at, 50s? Anybody remember? 1950s. Women's rights, not the greatest at that point in history, unfortunately. And so...
Wasn't she dead by the time we found it anyways?
So even if she's dead, if you've still discovered it, and it just wasn't publicized, and someone comes along and says, oh, look at that, well, I'm going to call that my work, that's a little sketchy. Okay? And so, yeah, there's a large kind of argument over who discovered what and who should have gotten credit. Ultimately, Watson and Crick are the ones that have been given credit for that. They definitely did a lot of work, okay, within that. So it's not to take away from their work. Rosalind Franklin really kind of provided the experimental evidence to really prove that we're looking at this. She should have gotten a little bit more credit, yeah. And then you run into citing sources as an issue. And then another name that popped up was the name Chablis, which I don't remember anything about, and apparently history is kind of forgotten. But he's also part of this discovery. So within this, what we end up getting is our strands running anti-parallel. And we can see that if you look at the top really, really carefully. We've got a 3' on one end, which is this kind of gold strand running all the way through, cycling around. Okay. So that 3' will ultimately run to a 5' end. So one of the things that's kind of interesting about our DNA single strands is that they do have ends to them, opposite ends. Okay. We have a 3' and a 5'. And once we hit that 5', we can now take a look at the other direction. We have the 3' running the other direction, okay, going back up to the other end of our 5'. So they run in anti-parallel directions. Okay. They partner according to the nucleobases. So G partners with C, A partners with T, and you end up getting these kind of flat ladder steps. So if we kind of uncoiled this, what we'd have is just a straight up ladder. We just climb the ladder. What ended up happening is due to the structure and steric considerations, those coiled around each other. Now a lot of that has to do with the backbone structure, with the connectivity on those sugars to your phosphates, okay? Kind of forcing that coiling structure. Within the base pairs, this is where it gets a little bit more interesting in how they go through and align. So every adenine is supposed to interact with a thiamine, every guanine is supposed to interact with a cytosine. Interestingly enough, what we're noticing is we have a pyridine with a purine, not pyridine with pyridine, not purine with purine. Why? Any ideas on that? So hydrogen bonding does form between these. That is absolutely true. And if this is all the information that gets stacked into our DNA, okay, then the question is, why do we always see pyridine, a pyridine with a purine? Why is it never pyridine and pyridine, or purine and purine?
Because you've got thymine binds to adenine and cytosine.
So that's ultimately looking at the pairings that we found, and we can address that next. Why is it always, make sure I got this, adenine to thymine and not adenine to cytosine? Okay. That can come down to our hydrogen bonding interactions, how they align. We end up with a more favorable hydrogen bonding interaction between adenine and thymine than we do with adenine and cytosine, which means we don't get an AC coupling, we only get an AT. Okay. But that still doesn't explain why I don't get A to G, or T to C, or A to A for that matter. What's that? Getting at the right idea, the way it fits in. What's different about those structures? 2 rings versus 1 ring. If I bonded 2 rings to 2 rings, what's going to happen to that distance between our sugars on the end? It's going to be bigger, which would then cause the DNA structure to bow out. Okay. Let's say we did do some adenine to guanines, and then we had the possibility for thymine to cytosine. Well, what happens with thymine to cytosine? It gets smaller in. Are we going to get that maximal interaction for hydrogen bonding? If one, we've got it kind of bowing outwards, which now means the next nucleotides on either side of it have to also have that. Okay? So it's not going to allow for that nice, perfect matchup. So a mismatch causes the DNA helix to bow outwards. Well, if it bows outwards, we're having imperfect interactions between those 2 chains. As soon as we have imperfection, what ends up happening?
All sorts of problems.
Problems because we're trying to fix it. Okay? Damage can happen where we've got imperfections.
Isn't that what we have, was it a replosome, which goes around and looks through all your DNA?
So we've got all sorts of repair mechanisms to prevent this from occurring, okay, or to minimize this type of occurrence. That is looking in our, actually the next 3 chapters, I think. What one was this? Was this 11, 9?
9.
So then it goes 10, 11, 12. Replication, I was getting backwards, transcription, translation, okay, for each of those chapters. We'll try and overall summarize those, but there's a lot of repair mechanisms in it, and that's actually a whole nother fascinating story, is that you're looking at 99.9% successful replication translation. Okay? That kind of lack of mistake is pretty insanely amazing. So we can't get pyridines next pyridines or purines next purines, because that changes the distance of those outer rungs of our ladder, which then prevents interactions from happening in between. Okay? So that would cause a disruption in the structure. That disruption in the structure would get picked up and try to get fixed. Okay? As a not so pleasant example, I think back when we were all teenagers and we found a zit. What did we end up doing with the zit? There's an imperfection in your skin. What do you try to do? You try to fix it. Okay? So that's the same thing that's happening here. If we tried to mismatch those, we now have a zit in the DNA. The DNA goes, oh, I need to fix it. Okay? We do get specific hydrogen bonding between our base pairs. Okay? So that's why we always see guanine with cytosine, and we always see adenine with thymine across the DNA strand. Would we see that in RNA? This is a general question from your biology knowledge. Would we see that pairing in RNA? No, because RNA is single-stranded, DNA is double-stranded. That's why we wouldn't see it in RNA, because there's only one strand. There's only half of the ladder in RNA, so we don't have to worry about that second strand interacting appropriately. Okay? So a lot of detail goes into the hydrogen bonding. As we can understand hydrogen bonding very well, you see positives and negatives. Okay? You get the general negative charge on your oxygen over here, which interacts with the general positive charge coming from your nitrogen over there, so we get a strong interaction. We get a general negative charge on that nitrogen, general positive charge on that hydrogen. So we can see that interaction. We want to cancel out that charge. Turns out that that's not the primary stabilizing factor when we're looking at DNA, which is kind of bizarre. Our primary stabilizing factor is this thing known as base stacking. Okay. This is going to be a stretch because we already know we've got issues with organic chemistry. Anybody remember the Diels-Alder reaction? And in the Diels-Alder reaction, you had endo versus exo attacks. So for those of you that remember this, you can be thinking about it. And we always ended up with the endo attack. Why did we get the endo result? Because of something known as pi stacking. Our base stacking is p orbital pi stacking. Each of those rings, okay, has a lot of p orbitals on it. Every one of those carbons, or nitrogens for that matter, or most of them, are sp2 hybridized. Right, which means there's a p orbital left over, which is going above and below the plane of the molecule. Okay. With those p orbitals, we can then stack the p orbitals on top of each other, and they can then stabilize each other. We're not forming an official bond between the p orbitals, but all we've done is kind of stack them on top. So as a general example, if we looked at a p orbital system, or a pi system, we'd have overlapping orbitals, overlapping p orbitals within our pi system. Okay, so there's one pi system I'm going to have to hold in one hand. If we take another pi system over here, the stabilizing factor that we're talking about is this. Okay, they can stack on top of each other. That stacking effect that we're seeing with our whiteboard markers is the same kind of thing that's happening within our DNA. So each of those nucleotides is running as a flat structure, almost like the tables. Our pi system is above and below that. When we take another table, we can stack it on top. Okay? And that kind of pseudo-p orbital overlapping is what's helping to stabilize that ladder structure. It's not a perfect overlap, because what happens is we take our DNA. Is it a perfect ladder? No, it spirals. Okay, so if you looked at a spiral staircase, you'll notice that in the center, or partly in the center, you get some overlap of each of those steps, but they aren't perfectly overlapped. It's that overlap that's ultimately one of the things that's contributing to the DNA structure. Okay. Take it a step further. All of those p orbital interactions tend to be very, very non-polar. Okay. P orbitals don't tend to have a polarity associated with them. If we look at our solvent in biological systems, the solvent is water, which is incredibly polar. So what are those p orbitals going to try and do? Get away from water as best as possible. You end up stacking them in between, so you get actually through the middle of your DNA structure a very hydrophobic region, which is kind of counterintuitive because you also have hydrogen bonding. Okay. But that's one of the things that's contributing to this. So that twist that we get out of it is trying to minimize its interaction with water. That's all that's really happening. Hey. What? Sorry, I thought I had a different figure. That's clearly way too far. Where'd my figure go? Well, that's a bummer. I'll just have to go back to this one. When we look at that twisting structure, you'll notice that we don't have a perfect coil all the way down. And what I mean by a perfect coil is that we get equal distances all the way through. The distance here is significantly different than the distance here. Okay. So what we end up having is something known as the major groove and the minor groove, okay, which could be nice if we think about replication options. If both of them were the same size, there's going to be no specificity between those two options. Okay. So if we have a minor groove, that information is now buried further inside the structure, and the major groove is now open, more accessible. Okay. So the more accessible region could be linked up with other enzymes or other processes to help open and now actually start to replicate your DNA. So we have to be aware of that major and minor aspect. Totally thought I had a better figure, but I guess not. Okay. So this is where we're minimizing our water interactions in that minor groove. You're shrinking that space down. We can move up to tertiary structure. So we've got ultimately our ladder form as kind of our primary, which we then twist into our secondary, and then we can continue to twist that, and that puts us into tertiary structure. So we're looking at two systems here. Okay? One of these is prokaryotic and one of these is eukaryotic DNA. What's going on? Anybody remember the difference?
Prokaryotic is circular.
Our circular DNA is prokaryotic, which I know is written up there, but might as well write it again. And our straight chain is eukaryotic. So what ends up happening is even though we've got these kind of tight coils within DNA, is that we can continue to kind of ratchet that down. So if we took the ladder, all we can do is just twist, and we form initially a relatively stable DNA molecule. As we continue to twist that, we end up doing a process known as supercoiling. That supercoiling works with both circular DNA and linear DNA. Okay? And what ends up happening is you end up forming these tighter loops within loops. Those loops are then further stabilized by different proteins within the nucleus. Okay? Typically, you're looking at your nucleosomes. Okay? So your supercoiling can occur in both species, or both, not species, divisions? What do we call prokaryotic versus eukaryotic? Types, classes, okay. It occurs in both ways. Okay. As we positive supercoil, we get one result. As we negative supercoil, we get another result. Okay. Which may seem kind of odd. Anybody used, well, most of you are probably old enough. You know the old phones that are on the cord? Picked it up, like the corded phones, and they had the coil on it? Anybody play with that cord on it? And either wind it tighter and start to see it loop back on itself, or wind it looser. That's what's happening. So as you coil the DNA more in one direction, you start to tighten it up. What is that going to do to the stability of the DNA? I know it's kind of an odd thought. If we tighten the coil, what happens? Is it more accessible or less accessible? Less accessible. I think I said that. So it becomes more stable. As we loosen the coil, what happens? Becomes more accessible. We actually make it reactive or potentially more reactive with other enzymes in the cell. So depending on what stage of life we're in, do we need some kind of transcription or translation or replication process? Then we need to unwind that DNA so that we can get at it. Okay, so you'll have enzymes that will go through and undo the supercoiling process so that we can get at the genetic information that we can then replicate or transcript for later processes. Okay. If we don't need to get at the genetic information, we want to protect it as much as possible. So that's why we'll coil the other direction to then trap all the DNA or the genetic information tighter in so that enzymes can't get through and activate it. Okay? The enzymes that are involved in this are typically your DNA gyrases, okay? Also known as your topoisomerases. Not that big of a deal, but it's terms that show up. Our quaternary structure is looking at our supercoiling process. So if we've coiled this up enough, we're going to end up getting different little pieces coming in to stabilize each of those coils. So our DNA coils can then wrap around proteins, and those proteins will then stabilize that DNA further, potentially also unstabilizing it. Not really address that right now, but you're looking at histones with different little pieces that come into it. Okay. As far as the exact role of the histones, one of our thoughts may be that it actually stabilizes the DNA and prevents interactions. That thought has been coming kind of into question because the histones can also then force that bend and cause things to open up and become more accessible, or allow for an enzyme to come in and bind, because now you're not binding to just the DNA, you're also binding to the histone. So you can improve interactions. So it is an active area of research. If you take a look, you can find nice pictures of these process or this coil of both the histones and your DNA. That's typically referred to as chromatin. Okay, so you can find pretty pictures of that, of actual chromatin molecules online. Denaturing DNA. All that's happening when we denature is that we're going to disrupt all of those kind of structures and take it all the way down to single-stranded DNA, much like we did with proteins. So when we look at our protein, if we denature the protein, all we're breaking is our intermolecular forces and breaking that into a single chain. When we're looking at denaturing DNA, that's all that's happening as well. We're disrupting all the hydrophobic, all the hydrophilic, all the hydrogen bonding interactions to get us to those single-stranded systems. Once we're now in the single-stranded system, we're now denatured. We can do all sorts of fun stuff with it if we wanted. Okay? All we have to do is add heat. Okay? We don't need to add any chemicals, because again, we're not breaking any bonds. All we're really doing is breaking those intermolecular forces apart. Okay? So heat works pretty nicely for this. DNA, because of all of those kind of hydrophobic and hydrophilic interactions running through the middle of it, through each of our individual single strands, once we remove that heat or remove the energy to prevent that interaction, it will coil back on itself. Okay? Because what it's going to try and do is minimize that hydrophobic and that hydrophilic system, so it will actually self-coil and renature, if you will. Okay? When we look at this process, one of the things that's kind of interesting, or we can look at different ways to observe this, we can look at ultraviolet light. Okay? We can look at the absorption at 260 nanometers. Okay? What's happening is that as we stack our base pairs, we're actually increasing our absorption. Okay? Why? We're maximizing the interactions between these. We've extended the chain long enough, and we can actually see a change in the absorption at 260 nanometers, because we're now changing those interactions between the individual nucleotides. So kind of a neat process. If we looked at our individual chains, and we said within this structure, I just listed off some of these, we had G, 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 G. Let's just say those are all G's. What would be on the other side? C, all the way down. If this was our sequence, it turns out this sequence of DNA has a much higher temperature required to cause it to denature. Seems a bit odd. Why would it have such a higher heat necessary? If you look at the interactions way back at the beginning, the hydrogen bonding interactions between adenine and thymine, there's 2 hydrogen bonding interactions. Guanine and cytosine, 3. So to break guanine and cytosine away from each other, we have to provide more energy to just cleave those 2 nucleotides away from each other. Okay? So we end up seeing a difference in temperature when we go through and do that process. Okay? The annealing is now just the reverse process. So you may hear of denaturation and annealing. Annealing is just now letting the two DNAs to come back together, or single-stranded chains come back together. When we look at ribonucleic acid, what are our big differences? What's the big difference between ribonucleic acid and deoxynucleic acid?
Only looking at one strand and finding
Single strand.
uracil.
We do have a change from thymine to uracil, and one strand, thymine to uracil.
It can leave the, isn't it RNA what leaves the nucleus?
RNA can leave the nucleus.
RNA cannot.
As a hint, it's a different sugar molecule. It's ribonucleic acid versus deoxyribonucleic acid. Okay, so our sugar molecule also changes in this process. So we're looking at just, or a standard ribose molecule. You're saying uracil pop in instead of thymine, and it's single-stranded. There's a variety of uses for RNA. It is primarily, or one of its uses acts as a transcriber, which means what we can do is convert the DNA into RNA as our carrier, and then transport that carrier information out into the cell to get translated for instructions. Okay, why might we want this as a biologically advantageous thing? Why not just go directly to the instructions and just use the instructions? Why not use the DNA as your template for everything?
I don't know if you screw it up.
Gone to like an amusement park, you got a map trying to find where you're going, right? You pull it out, you keep pulling it out throughout the day. Eventually what happens to the map?
It's all messed up.
It's all messed up. Okay? So what we're trying to do is maintain the integrity of our instructions. Okay? That's the idea on the DNA. Here's our main instructions. Let's transcribe that out to a copy. Now that copy can go do all the work it needs to. If it gets destroyed somehow in that process of making it out to where it gets copied, oh well, I still have my main genetic information. Okay? So I can always make more. So that's one of its primary roles. So DNA is going to end up dictating the RNA sequence. So ultimately, they're just mirror images of each other, okay? Except that you're exchanging thymine for uracil. So your RNA sequence has uracil. There are lots of different types of RNA. There are the three primary ones that you've probably all heard of before. You've got your transfer RNA, your ribosomal RNA, and your messenger RNA. Okay. You get variable sizes with your RNA and mRNA, and then your tRNA is relatively small. Each of these have different roles. So your tRNA is known as transfer RNA, because what it is transferring is it's moving amino acids to your M and R RNA to synthesize proteins. Okay, so all it's doing is facilitating protein synthesis. Your rRNA is ribosomal RNA. What this is doing is actually making your protein. It's the enzyme that's actually facilitating this transfer and putting all those amino acids into the proper sequence. Well, how does it know what the proper sequence is? That's messenger RNA. So messenger RNA carries that information. The ribosomal RNA reads that off and matches that up with our tRNA to go through and do our protein synthesis. So those were our primary RNA types. Turns out there's a whole bunch, or a slew of other possible RNAs. So some of the common ones, sRNA. This is a small nuclear RNA, hence the SN. It processes the mRNA before it leaves the nucleus. Okay, so our mRNA has a bunch of extra stuff in it that we need to get rid of. Okay, so the DNA has a bunch of extra information. When it gets transcribed into the RNA, we now have that digital, or what is it? High fidelity copy. Just saying it's the same. Okay. It turns out there's sequences within that that we don't need. Okay. So that has to get processed and removed before we actually go out into the ribosomal RNA to get turned into proteins. The enzymes, if you will, that process that, that's your sRNA. Okay, your sRNA, along with some other proteins. That complex is known as your SNRP, your SNRNP. Okay, so that is, I believe, your small nucleic ribonucleic proteins, maybe. Okay, so it's those complex with some proteins to help process your mRNA. Your siRNA are interfering. So your S is again for that small, and they're interfering, meaning what they're doing is they're getting in the way of some process happening. Well, they're still they're still genetic information. So what's happening is that small interfering RNA will get in and bind to different sequences, either in the mRNA or in, say, DNA, and prevent certain processes or prevent certain genes from being expressed. Because if we now imagine you've got a train, okay, running along the train tracks. If you throw now a brick in front of it, that can derail the train. That's ultimately what's kind of happening with your siRNA. Okay, you're putting something in to prevent the train from passing through. Your transfer RNA, we've got some kind of neat pictures coming out of it. General, smallest, got a whole bunch of random stuff on it. You've got lots of hydrogen bonding within the chain. So remember we said RNA was all single-stranded. This doesn't look very single-stranded. When you look at these pieces, a lot of that looks like we've got double-stranded RNA. Well, just like DNA, we can sync up with another strand. tRNA is still single-stranded. If you look really carefully, what's actually happening? The strand folds back on itself. Okay, so it is still single-stranded. It's just the chain is folding on itself. So we still maintain that single-stranded action even though we've got these kind of weird cross interactions. That gets you a different shape out of your tRNA molecule, and it ends up generating these kind of weird alpha helix types shapes. Your tRNA does need to get activated, so I don't know how much detail we're actually going to go into on this when we move further on. Your tRNA gets processed by this big long enzyme named transferase. What that's doing is going through and adds the nucleotide CCA. That now activates the tRNA. It's now good enough, or it's now qualified enough that it can go out and act as tRNA would, start picking up the appropriate amino acids that it needs to go through and transfer. Okay? If for some reason there is something wrong with that tRNA molecule, say it was processed by the DNA and there's something wrong with it, it's not folding appropriately. Well, that transferase comes up, recognizes that it's not correct, that there's something wrong with it, and tags it instead of with CCA, it double tags it. CCA, CCA. What is that doing? It's now tagged that enzymes now look for this signal and destroy it. Okay. So all we end up doing is then taking that all apart so that we can potentially rebuild it later on. Okay. So you can almost look at it as a sequence of Lego blocks. Okay. We put something together and realize that we built it wrong, so we take it now apart and try and build it again. Okay. Your ribosomal RNA, roughly 60 to 65% by weight. We're looking outside of your nucleus. What these are going through and doing is processing your mRNA to build your protein. So this is kind of a neat little picture. This came from UC Santa Cruz, where they had actually solved the crystal structure of this. If we look really careful, in fact, do I have a link? No, I don't see a link. I think, if I remember correctly, you can actually get this crystal structure online and you can rotate it and change it around. See the yellow sequence running through the middle? That yellow sequence is, I believe, a protein strand forming. Okay. So they were actually able to crystallize it in its sort of mid-synthesis process, which is kind of neat. It is composed of two primary units, a 30S and a 50S. Anybody know what the S is in reference to? Probably just told typically it's just 30S and 50S, which is kind of where we're going with it now. The S has to do with its centrifugation gradient. So where did that when you centrifuged a sample down at very high speeds, where did that chunk come out? Okay, at what level within that centrifugation process? So it has to do with its size, or ultimately its density for each of those. The 30S is further subcategorized down into a 16S RNA and then 21 different proteins as part of it. And then your 50S has further decompositions down as well. Okay? Ultimately, all you really need to know is your ribosomal RNA is the one that's actually processing and making your proteins by reading mRNA and bringing in the tRNA. When we get into the translation, I think I have a couple cool videos. YouTube's full of nice little videos that show these kind of processes. So we'll look at those a little bit closer. Your messenger mRNA is the one that actually carries the information out to your ribosomal RNA and then gets turned into your proteins. So it's not quite sure how we want to call that. It's your information that's getting processed. Okay? It tells the RNA what amino acids we need to bring in. Okay? It typically has a very short lifespan, which seems kind of odd. Why might it have a short lifespan? It's just meant to transfer information. But if it's an important protein, why, or information for a very important protein, why would I want the mRNA to decompose quickly?
So it doesn't keep getting synthesized?
So it can be a synthesis problem. We don't want to keep synthesizing that protein. And remember, we have to have this homeostasis effectively within our cells, which means we don't want to keep producing a protein if there's nothing for it to do. Okay? Why? You've got one set of Lego bricks. Okay? That's all you've got. Okay? If you keep synthesizing that protein, you've now put a whole bunch of amino acids into a protein that's not getting used. What if you need those amino acids somewhere else? You now don't have that information. What if you're going through a replication process? You need nucleotides to double the DNA. If you've got your mRNA held up, holding on to nucleotides, you can't process. You don't have the pieces to continue that processing. So we have typically very, very short lifetimes on your mRNA. We want to get that information out, get it transcribed or translated as quickly as possible, and then move on. mRNA is synthesized from DNA, random facts. In its processing, remember we talked about the SNRPs and your sRNA. It gets processed in the nucleus before it leaves the nucleus for your ribosome to get processed. Okay? There are two parts of that. You have exons and you have introns. Your exons are the good things, the introns are the bad things. Okay? So your small nuclear RNA and those proteins will go through, read through it real quickly and say, hey, this is a bad section, I don't want this section. Facilitate the removal of that and then linking the two pieces back together. Okay. That is your quick overview of all nucleotide information. Like I said, it was quick. So which of the following groups is not found in nucleotides? All of those are found in, that should say nucleotides, in your nucleotides. Okay. Remember, your sugar is your ribose. Your purines and your pyrimidines are the nucleobases. The phosphate is necessary to make it a nucleotide. That's what that name change comes in. Which sequence of DNA should result in a higher DNA melting temperature? So in our denaturation process, what we're looking at is which one has the highest intermolecular forces. The one that's going to have the highest intermolecular forces is the one with the most C and G units, because you have 3 hydrogen bonding interactions as opposed to 2. Which of the following is false for mRNA? Short lifetime, must be processed before leaving the nucleus, contains exons, moves amino acids for translation. False. D.
Transfer.
Okay. It does not move amino acids. tRNA moves the amino acids. rRNA processes the mRNA. The mRNA just has the code. Okay, so it just runs the code straight through the rRNA, and the rRNA brings in tRNA to bind in and generate your translation process. Which of the following statements is true only for DNA? This is one of my favorite questions. I think it's actually kind of challenging.
B.
It's A. Why is it A?
Just when it's transcribed, it's the opposite.
Nope.
Because of the way it spirals.
What do you mean by the way it spirals? You got the right idea.
By the way that it bonds with the second strand.
The second strand is the part that's important. In rRNA, there's only one strand. Okay, we don't need a balancing out of G and C. DNA is double-stranded. You will have equal amounts of G and C. Okay, because if you have G, what's the second strand have? C to balance it out. Okay, so when we're looking at DNA, because it's double-stranded, you have equal percentages of G and C. Okay, it was kind of one of those things like, that was really obvious. Why did I not think of that? Okay, so it's going back to the overall structure on that. We still have our sugars connected 3' and 5'. They're definitely through that same phosphodiester. The pyrimidine uracil shows up in just our RNA. And then adenine shows up in RNA and DNA. Okay. And so what the question was asking is true only for DNA. That's only going to be A. Which of the following are examples of RNA? Are that question? This was interfering. That says interfering. Let's try that again. Uh-oh. 2 R's? Let's just do that. Interfering RNA, transfer RNA, and ribosomal RNA. All are examples of RNA. RNA. I keep throwing in that second R. Yes?
After, sorry, I'll ask you in a minute.
Okay.
Go back to the DNA slide.
Okay. Any questions about the questions that are on there?
Or, yeah, that one.
That one?
RNA's slide. Do the numbers change then? The 30s and 50s?
Yeah.
So those aren't constant. Just the S part's constant. It just
Yeah, it's between prokaryotes and eukaryotes, I think those numbers change. So the S is just referencing when it gets, when you can isolate that part of the RNA. And so it's sort of relative to its size, because ultimately all of them fold up on each other, so their densities are roughly similar. Wait, no, that can't be right, because it separates based on density. Yeah, it separates based on density. It's a centrifugation process. So they'll go to different levels within your centrifugation. So I don't, and it turns out that the larger the S value, typically the larger that sequence is.
That makes sense. Just from 1 to 1 to 1, you're never really going to see a 50-30, maybe a ratio like that. But
Yeah, so it depends on the life form, where it's coming from. And I believe it's prokaryotic versus eukaryotic.
thank you, sorry.
No, not a problem. So what I'm going to try and do is, because it's roughly 15 minutes left in class, is kind of whip through what I would kind of expect out of your presentations when we talk about virology. Before we do virology, I wanted to make sure that we're all aware for our 10 to 13, chapters 10 to 13, is known as your central dogma. It's looking at how your genetic information makes it into, I guess, the real world. So you've got your DNA, which can be replicated. So that's chapter 10, I think that's chapter 10. There's too many chapters there. I think it's chapter 10 is your DNA replication. You then have transcription. So transcription is the process of going from DNA to RNA. Okay? RNA can be replicated as well. Okay? But we're looking at our primarily just going left to right. And then from your RNA to your proteins is translation. So you've got those 3 primary processes. And we'll kind of look at each of those processes to certain aspects on Tuesday next week. And I've got a little pseudo game that we'll try and play with replication. Kind of look at what aspects need to go into that. Okay, so I just want to make sure you guys are familiar with those terms and what they're referencing. And what's going to be of particular importance here is actually this reverse transcription. There are certain processes where you can then go from RNA into DNA. Okay, as we already kind of discussed, that's not really a good thing to do because our DNA is our primary store of information. So it seems kind of odd of why we would ever shift from RNA to DNA. What the heck? So viruses, these are in general our pathogens. They can be, and what they will do is go through and infect bacteria, plants, animals. They pretty much infect anything out there. So all they really have is a bunch of genetic information that they just design themselves to just go through and replicate. It's kind of their goal. They can be deadly, but not always. Okay? Really all we've got with those is that they are small particles that hold nucleic information, genetic information, that then gets replicated by a host cell. So they infect, force the host cell to replicate the virus itself, that same genetic information. It then propagates itself through its, I guess, disease, if we want to call it that. The entire particle is known as your viron. It's composed of a couple different things. So first off, we definitely need the genetic information stashed within it, but we need something to protect it, because that genetic information on its own, once it's outside the nucleus, right, which in the case of a virus we don't have that, it can get processed and destroyed by just natural things and natural enzymes. So what we want to do is somehow get that genetic information into a point where it can be replicated, which means that our virus needs to somehow get that genetic information through the cell membrane and into the nucleus. To go through and do that, it's got a bunch of other little pieces behind that. Okay, so we've got our capsids, your nucleocapsids, membrane envelopes, and protein spikes. All of these have various roles to go through and run such that we can get that genetic information through. So we've got kind of our nice little picture of everything. You've got the membrane on the outside to kind of keep it trapped. You've got the spikes on the outside. Those are going to help inject. You've got your capsid protein coat, which is now protecting the nucleic information on the inside, okay, your genetic information. So if it's going to go through and kind of propagate itself out, this genetic information has to have everything, or have the information to code for everything on the outside of it, okay, and ultimately for the genetic information in the middle. Okay. So in general, what ends up happening is it binds to your cell membrane, drops the spikes in. Once the spikes have actually adhered, that's going to facilitate the injection, almost like a syringe, of the genetic information into the cell. Once it's in the cell, now we've got through kind of our primary goal, and we can start to get that shifted around where it can then get processed either into forming proteins. So we can then do the transcription, if we're starting with DNA, into RNA. Then the RNA can be converted into proteins, which then can generate all the pieces that we need to reform a new version of the virus. Okay? So ultimately that genetic code comes in and then has to deal with different processes. Okay, so we talked about one aspect, going through your transcription so that we can get all of the protein pieces necessary for the virus itself, or its viral structure. But then we need all the genetic information replicated as well. Okay. Depending on how it's transferring that information, it can transfer it as DNA, in which case it's got to go immediately to our nucleus, or the nucleus of the cell, so that it can get translated, transcripted, and then translated, okay, and replicated ultimately outwards. Or it can come in as RNA. And if it comes in as RNA, that becomes a little bit more challenging. The RNA, for sure, can go through the transcription or translation process. Okay. But RNA doesn't incorporate into the DNA. We can't now convince the cell to make all of our stuff for us. So the virus has to have some way to convert the RNA into DNA so that it can get incorporated into the cell's structure. That is your reverse transcription. Okay. A virus that does this is a retrovirus because it's reversing your central dogma. So what will end up happening is there are individual receptors on your virus that match up with different receptors on the cell membrane. Once it's identified a cell and is locked on, it can then shift and inject its little spike, which then can pump in all the nucleic information into it. Okay? So we get all sorts of little fun stuff like that we can envision. So your retrovirus has a whole bunch of different stuff coded within it. Okay, so we've got your typical retrovirus. You've got your standard proteins, okay, to code for the outside. You've got your reverse transcriptase. Okay, remember, we're talking about the retrovirus. We need this. Okay, the reverse transcriptase is to take it from RNA to DNA. So that has to show up in your retrovirus. And we'll also have genetic information for the envelope. So how do you rebuild that viral structure? There are some other viruses they list out there just so that you can see it. The important part there is your reverse transcriptase. The life cycle, inject, get it into single-stranded DNA, get your single-stranded RNA turned into double-stranded DNA. Once we now have the DNA, you now need to get that put into the genetic information of the cell, so the cell now replicates your information. Okay? That involves doing an integrase. So there's an integrase enzyme that is going to take the double-stranded viral DNA and insert that into the double-stranded host DNA. Okay? So that integrase is kind of a neat little process. It helps chaperone into the nucleus. It goes through and cleans some of the stuff up, and then it incorporates it into the host. Once in the host can now take over and do all the hard, busy work of replicating the virus. So the part that I thought was neat was looking at the integrase. The integrase is composed of 3 primary regions. At the N-terminal end of it, you have this HHCC, zinc binding domain. What is HHCC? May seem kind of weird, but think back to what we might see within a protein. The symbol for histidine was H. So you have a histidine, histidine, cysteine, cysteine bonding pattern. Both of those, or all of those amino acids, will link up and bind to a zinc atom. So we've got 2 possible ways we can generate this. Okay? So different shapes come out of this. These are known as zinc fingers, okay? Because they generate these kind of weird finger-looking things. And they're coordinated all because of that zinc holding that protein down in shape. Okay? What's kind of neat about these zinc fingers is that these can then wrap around your double-stranded DNA. Okay? So they can hold on to that DNA molecule by sitting within the major groove of your DNA strand. So those alpha helixes fit almost perfectly within the major groove of your DNA strand. Okay? So they can help carry your viral DNA. After that, we've got 2 more pieces. You've got a catalytic core. So this is where the chemistry is actually happening, where we're transferring the viral DNA to the host DNA. Right? We'll talk about the catalytic core in just a second. At the C-terminal end, you have another DNA binding zone, which is known as the SH3 motif. Okay? Remember we talked about when we looked at protein structures, we had alpha helices and beta sheets, okay, and beta barrels. There are other motifs. That other bonding motif that we saw here was a zinc finger. Okay? Another motif we have here is this C, or it's not a C, SH3 bonding motif. This is your SH3 pattern, which looks an awful lot like those were our beta sheets that have then formed a barrel. We have a beta barrel. Okay? That's also going to help bind our DNA. Okay? So we've got our overall integrase protein shown in that last image here, okay, which has now everything incorporated into it. So you can see the different binding sections. You've got these alpha helices, which are probably due to your zinc fingers. And then it looks like we've got probably 4 SH3 bonding motifs within it as well. So the whole point of the integrase is really to wrap and fold itself around the DNA in 2 versions of DNA, your host and your viral DNA. Once it's gone through and done that, it then has to catalyze the reaction. So what it's got to do is take your host DNA and incorporate the viral DNA. Okay? So it has to go through and break bonds. There's a variety of different types of integrases. You can refer to the serine integrase, the tyrosine integrase, or there's actually another one that's just no covalent catalysis. Well, no covalent catalysis is boring, so let's ignore that one. What do the serine and tyrosine both have in common? No.
It's close.
Yeah, there is a ring in tyrosine, I will grant you that, yes. Both of them have that nucleophilic oxygen. Okay. So the serine or the tyrosine have a nucleophilic oxygen, which can then go through and attack the phosphate backbone. Okay. What that will end up doing is ultimately cleaving the phosphate backbone, and you now have a new 5' and 3' end, which you can then coordinate to the new DNA structure, or your viral DNA structure. Okay? So your serine and your tyrosine are both there as a nucleophile to go through and physically break open that bond. And you now form a covalent linkage to the enzyme itself. And of course, they're going to require all sorts of other things. There's this reference of ATTP in both cases. It took me forever to find out what that meant. Anybody know what it means? Good, because then I'd feel like an idiot. Okay. ATT is standing for attachment, and P is phage. Okay. So it's the sequence of DNA of where your integrase actually wants to cleave open the phage DNA so that it can insert it into the host DNA. Okay, so it's a DNA sequence that it's recognizing for cleavage to insert. Okay. So your serine integrase apparently goes after a short sequence, and the tyrosine goes after a long sequence. One thing that we could potentially guess out of that is, since we're looking at a shorter sequence of DNA to interpret or insert, we don't need as many cofactors. We don't need a bunch of other stuff to help facilitate that insertion. If we're looking at a longer sequence that we have to deal with, you're probably looking at a bunch of other cofactors being involved in that. And we just looked at that, which I was pretty sure I double included. So then we can jump to the exam questions on this. Right, so you get the general idea of what's going on the presentation. Quick overview, talk about some structural aspects. I think the structural aspects are kind of cool. And then getting into the kind of organic aspects. What's going on with the chemistry behind it. Because we are out of time, I'll try and real quickly bubble these answers in. Integrase is used by viruses to viral into host, not host into viral. Not a step of the retroviral infection. You are not transcribing DNA. Okay? Viral DNA into host DNA is an insertion. At best, you're looking at a replication process, not transcription. The transcription is viral to DNA, or RNA to DNA. Retroviruses store their genetic information in RNA. So that's the retro aspect. You're reversing your standard process. True, false, viruses are always deadly. False. You can get the cold. The following are secondary, tertiary proteins, except for secondary or tertiary protein structures. And I think I can already tell you that if I use this question, I'm going to change that to motifs. Except for all are valid examples. So your alpha helix and your beta sheet are the standard ones that you've already seen. The SH3 and the zinc fingers are the new ones, or new bonding motifs that we hadn't talked about before, but show up in the integrases. Okay? If you've got questions about